Let's record. Now let's share. There we go. Okay. Oh my goodness. All right, crank this down. Turn on one of those lights. All right. Okay. So the good news is, like I say, this first exam, I won't make it right after Labor Day. It'll be a week from today. So I'll put that up on Canvas as an announcement, because I know usually after about a week, the announcements kind of slide down your screen. So I don't want to put them up too early, but we'll have our exam and it'll cover chapters 1 through 3. Now, chapter 4 actually really ties in with that, with chapter 3, but you know, I don't want to give you 4 chapters.
Accessibility options.
Thanks.
Use the tab key.
No, stop. Is that yours? Oh, yeah. Okay. Yeah. I was like, what in the world? Okay. Let's make me, there we go. We'll do that. Okay. And now I can do that so I can see everything. Yeah, awesome. Okay. Yeah, I will say actually having your computer be able to read to you was fantastic when I was doing primer design and reading off the sequences. So I'd have my computer sit there and go A, C, G, G, D, and so I could just check it off. Yeah. Anyway, so jumping into our proteins, we will be maybe. Come here. There we go. Yes. There we go. Talking about the three-dimensional structure of proteins in here. So this is actually not a terribly long chapter, which is good, because when you get to 5 and 6, it starts getting more and more and more. So I'll try not to go too fast in this one, but don't hesitate to slow me down. So our big thing here is we just want to learn about the structure and properties of our proteins and how our peptide bonding, you know, we take all these little individual amino acids and we hook them together and we get a big giant protein that has a particular shape and a particular function in there. And there are patterns that we can kind of suss out. There are different levels of structure. We'll kind of talk about those. And we'll talk about how we actually get these proteins to fold and how we can denature them and hopefully renature them. Okay. So our structure, we have this three-dimensional conformation. So if we think of organic esters, things like polyester or rayon or something like that, nylon, we typically get long fibers. Okay, most of our proteins are not going to be a kind of linear form. We do see some that have a linear form, like actin and myosin, but most of the ones that we'll be talking about are going to have a more complex three-dimensional structure. The globular ones, basically, a lot of them will look like blobs, or blobs with legs. And this whole structural component is necessary for the protein to fulfill whatever function it has to have. Okay? Even if we look at something like collagen, as an extracellular matrix protein providing structural strength, so for like your knees, you'll see it'll have all these kind of branches that come off of it, and the branches will have branches, and those branches will have branches, and those branches can maybe soak up water to like allow your knees to flex or something. Okay. But our structure of our mature protein, once it's made and doing whatever it is that it's supposed to do, we call that its native fold, okay, or its native structure. There will be a large number of favorable interactions between the various groups within the protein that hold it in that position. So for those of you who will be taking senior seminar in the springtime, there's one professor that always asks a question that goes back to basic chemistry, and the answer is always hydrostatic interactions. So what stabilizes these proteins? It's a lot of hydrogen bonding. There's just a ton of hydrogen bonding. These are weak little bonds, but when you have thousands and thousands of them, they help stabilize this overall structure. You'll see salt bridges and other stuff in there as well too. But we have so many of these little hydrogen bonds that help stabilize that structure. And the key is to get there, we have to overcome entropy. Because we do want everything just kind of like flying around like crazy, like our wacky waving car lot advertising guy, or let's say a thing there. That's a lot of entropy in there. We need to overcome that entropy and put it in the specific conformation to get to our native fold. And we do that by making these favorable bonds. Okay? We lower the energy that way. Okay. So let's talk about some favorable interactions in our proteins. We've got the hydrophobic effect. We kind of talked about that. Again, see how this ties in with chapter 3. If we put all our hydrophobic nonpolar parts in one section, we free up more variability for the water molecule structures to have. So hydrophobic effect helps, is a favorable interaction to overcome the decrease in entropy. Hydrogen bonds, massive stuff going on in there. Our London dispersion, again, our weak interactions in here that just kind of hold things stable. Our atoms kind of want to be near each other. And then we'll have some electrostatic interactions as well too. We can have like our permanently charged molecules, our stable charges that are in there. They can interact with each other over much longer distances and help stabilize the overall structure. We can also see salt bridges, which is basically where you have closer plus and minus bonds that actually kind of stabilize each other, hold them together. Okay. So all of these add up to provide energetically favorable interactions. We think of our total energy. These are going to be our interaction ones as we decrease entropy. It's got an energy cost. This is where we make up that energy cost. It's kind of like, I put in double paned windows so I can turn the AC up to like 60 now. That's my goal. Okay. All right. So that's a lot of words, but sometimes maybe a picture is more helpful. Come on. And then we go to it. So let's go back to the picture. There's the picture. There we go. All right. So I like this because this really describes, the essence of this chapter. We start off with our primary structure, which is just our sequence. We have our sequence data. Okay. If I give you the sequence data for a protein, the odds are you're not going to be able to figure out what its structure is. There are actually ongoing contests between modeling. I got to actually observe this when I was a grad student, where they're given a sequence and they need to figure out what the actual structure is. And the closer the models get to the real thing, that's who wins the prizes. But that's our goal, is to be able to take the primary structure and predict the final mature structure. Okay. But a lot of these little segments in here will adopt specific structures in here. We'll talk about alpha helices and beta sheets, as well as random coil. And we'll see these little these can come together to make motifs as well too. But this is our secondary structure. It's just a region that has a particular common pattern. Alpha helices, beta sheets, and random coil are the three that we'll talk about the most. When we have one single chain and its overall three-dimensional structure is our tertiary structure. So this is one peptide and just kind of folds around this way. So in this case, this might be myoglobin, which is made up of one peptide strand. Okay? It's got a heme component in there, but it's just one peptide. So every protein has primary, secondary, and tertiary structure. Proteins that are made up of multiple subunits, multiple peptides, will have a quaternary structure, which is how all the peptide subunits fit together. Okay, I always like the cytochrome BC1 complex in the iron sulfur protein because it's really an 11 peptide group, but it's a dimer of those two 11 peptide groups. So you've got 22 peptides in there that fit together. So that's clearly a more complex structure than if you just have one peptide by itself. Okay. So primary, secondary, tertiary, and quaternary structure are good kind of definition things. Or what's the difference between this and this? Might be a question I would ask. I get here and my voice gives out. Yeah, so can't talk too much, right? Okay. So let's look at these in a little more detail. Our primary structure is going to be the emphasis of our peptide bond. So we kind of talked about how that peptide bond takes place in chapter 3. You're going to have that condensation reaction where you're going to lose water in there. But let's talk about the properties of when that happens. We actually end up making this resonance structure in kind of a hybrid between two different structures. And that causes some pretty unique things to happen. What we end up seeing is that our peptide bond is less reactive than other esters because it's more stabilized by this resonance position. Also, the peptide bond ends up being very rigid and quite planar. So we have this illustration where we talk about having like different plates that are connected at the alpha carbons. So here's our peptide bond, here's another peptide bond. So we essentially have this series of linked plates that will fold around each other. We also are going to see this large dipole moment in our trans configuration, and something like 99%, greater than 99% of the peptide bonds will be in the trans configuration. So let's look at this pictorially. Here's our peptide bond as we commonly draw it, okay? But we're going to see what can happen. We can have this nice little resonance structure here where our bonding pair here can kind of flip down from the carbon-oxygen to the carbon-nitrogen, okay? So now we have a double bond here. And it's going to flip back up. So this is our nice little resonance structure. When we do get this down here, we end up with a negative charge up here and a positive charge stabilized here. So there's something funky going on there. But we end up with this stabilized charge in there, so we get this nice little dipole moment, okay? Because we have this resonance structure, again, we have partial double bond character. So that partial double bond character is what's going to make it very rigid and very planar. So we have this nice little planar section in here. Okay. All right. Okay. All right. We can also kind of get a little stabilize our we can put a lone pair on the nitrogen and have a carbocation there as well too. So that's a third possible resonance structure there. All right, so the plane allows for partially free rotations, and the slide after this one is going to illustrate this better than my little hand motions here. But so we have this rotation. The rotation around the peptide bond is not permitted, so that carbon to nitrogen linkage, it's not going to rotate. It's going to be stuck in that planar form. However, we can rotate around the alpha carbon. Okay. So we can look at where the alpha carbons are connected. You can have your phi around the amide nitrogen bond to the alpha carbon, and your psi from the alpha carbon to the carbonyl. All right, so what does that really say? Really, we just have these two bond angles, these two angles of rotation. So if we look at these, all right, we can see what's going on here. All right, so here's our carbon alpha nitrogen with our phi and our psi. All right, to the carbon. And so essentially we have these nice little plates that are stabilized here, and then they can rotate where they're linked at that alpha carbon. Okay, so the alpha carbon is free to move, or the plates are free to move around the alpha carbon. Our alpha carbon also has the side chain on there. So we essentially have these rigid plates that can rotate around the alpha carbon, and whatever the R group is going to determine a lot of the structure that's involved in here. Okay. So if we have a big bulky side chain here, we're going to inhibit the rotation of these plates. Okay. So we can talk about allowable rotations. Right? That gets us into the idea of the distribution of the possible phi and psi angles that are in there. Okay? We have to watch out for steric crowding. We obviously can't fold them back over on top of each other. It's kind of like me when I try and do yoga. It's like, oh, nope, can't do that. Because say after like 20 some years of martial arts practice, yeah, I went to try yoga, and it was weird. It was like an hour-long warm-up where we never got to the actual martial arts. But there were some things in yoga, it's like, oh, this is really easy, because I've been doing a similar stretch for 20 years. And then there are some things where I was like, what am I doing here? So, so there are some allowable stretches, and there are some that are just not allowable because, I'm old and I don't bend that way. All right. Okay. And it's crazy when you have your kids who are like, oh, let me do a split. Daddy, can you do a split too? No. No. Not even when I was young. All right, so some of these combinations are going to form favorable H bonding interactions. And so those are going to tend to stabilize the structure, and that's going to, again, help stabilize our overall three-dimensional structure. What we can do is we can come up with something called a Ramachandran plot that shows the distribution of angles that are allowable. And there's a lot of math in this. We could go into that in great detail, but then we'd never get through this semester. So we'll just kind of show you some allowable angles. What's in blue are our typical allowable angles. White, typically not allowable. There are some exceptions. You can induce some strain, or you can have some unfavorable reactions that are compensated for elsewhere. But most of the things we're going to see are going to fall in these ranges. Okay? We can further define some of these ranges for, say, right-handed alpha helices and left-handed alpha helices. This is almost kind of like a mirror fold of each other on the angles of rotation in there. We can see some parallel beta sheets and some anti-parallel beta sheets. Our college and triple helix falls in here. We've got all sorts of stuff in here. But most of the stuff in white is, the forbidden zone. So if we actually look at some plotted ones in here, if we look at glycine, you can see glycine is kind of all over the place. It's mostly in here, but there are a few that are out here because glycine with that side chain has a lot more freedom of rotation. So it can actually move around in places that maybe it shouldn't. Another residue would be prevented from moving. Okay, so again, the whole idea there is if we look at these rotation angles, that will tell us whether we're making favorable or unfavorable interactions. And favorable ones we're going to define as being allowed, and that can help us understand and predict structure, okay, from our primary sequence, our primary structure. Our secondary structure is going to talk about just kind of like localized little areas. Okay, it's kind of like if you're driving down the road and there's a little park, or you come to a roundabout or something like that. There's a little localized structure there. Okay, so this will be, I guess, our local spatial arrangement is another way of saying that. There are two very common ones: the alpha helix and the beta sheet. Our alpha helix is going to be stabilized by hydrogen bonds between different nearby residues. Our beta sheets are going to be stabilized by hydrogen bonds between different segments of the protein that are not necessarily nearby. They could be pretty far away. And then we also have irregular arrangements, which are called random coil. Okay. So what do I want to say about this? Okay. If we look at some typical angles here, I want you to know what the phi and psi angles are, but I'm not going to ask you for like this type of data in here. That's too much at this level. Too much. If you go into like protein folding, then yeah, you'll dive into this. Got to tell you my little aside, since I'm trying not to go too fast here and slow down. I did molecular modeling as part of my graduate work. Molecular modeling is now very well established, but this was fairly early on. And basically you had people who did benchtop work and people who did theoretical modeling work. And there wasn't much talk between the two. So, if I overcome my great humility that I have and actually like brag about myself, I was one of the first people to try and bring the two together. My great humility. But there was a lot of resistance. It took a couple years to get our first paper published that looked at both of these. I don't remember what I was going to say about this story now too. Blah, blah, blah. Oh yeah, these angles. So yeah, so I would even get some resistance from the department. So I'd present my work and people would say, well, what's the equation for how you're modeling this? So I'd get that, and it's pretty crazy. It's complex. It's this something that, the computer does, and I trust it. But I got that question like 2 or 3 times. So I'm like, fine, you want to see that? So I gave them this 11 variable equation, which was just ridiculous. I could have probably explained 6 or 7 of them in there. The other 4 or 5, I'm like, yeah, I'm not quite sure what these are, what these do exactly, but it was all in the modeling in there. And the best part was I showed that equation, talked about like the first few pieces of that equation, and then glossed over the next one, and like no one ever asked me about the equation for modeling again, because I was the modeling expert in my department. And honestly, I didn't know that much about modeling, but I knew more than everybody else in my department. So then people started asking me for advice, and I'm like, let me send you to somebody who really knows what they're doing. All right. Anyway, so the point is, if you know enough, then you can facilitate that knowledge. You can figure stuff out, and you can BS people. And I should go into politics, right? All right. Anyway. Oh, let's not be cynical this early in the semester. Okay, so let's talk about the alpha helix. All right, so our alpha helix is this nice little helical backbone. We've got hydrogen bonds in here. So basically, we kind of wrap these this chain around a nice little spiral, and our residues in purple here are kind of pointing out at an angle, and hydrogen bonds between the various amino acids can stabilize each other as they kind of go up this spiral staircase. Okay, so we'll typically see there are, I think, something like 3.4, 3.6 residues per turn. Okay, so as I go around this turn, 360 degrees around, as I wind up that way, we'll have 3.6 residues. So if I have 10 turns, there'll be 36 residues in there. And I'll see that maybe this fourth residue past this first one will be in a position to form a hydrogen bond. So it's kind of like the risers in a spiral staircase that helps stabilize the steps that are next above it. So that's what we've got going on here. The peptide bonds are roughly parallel with the helical axis. So they're kind of, our plates are going up the sides as well too, all right? And the side chains typically point out they're a little bit more perpendicular to the axis. Not completely perpendicular, but in general, they're poking out like the blades on a fan, okay? We can have right-handed helices and we can have left-handed helices. You guys have physics and do like your vectors and everybody sits here and raise their hand, right? Remember that? Does that sound vaguely familiar? Hopefully. We can do the same thing with these. And then the best part is we can have stuff like, we can take 3 right-handed helices and wrap them around each other and make a left-handed superhelix, and that's collagen. We'll get to that later. But our right-handed helix is just talking about, the way that our protein is going to go from our N-terminus to our C-terminus. We're going to wrap around it this way. I think of this, and I think most of the things we end up talking about tend to be right-handed, but it's kind of like, there's a castle in Scotland where it was built by a family that had some genetic predisposition to be left-handed. So most of the castles had this, I think it was a right-handed staircase so they could fight up and down the stairs and have the advantage of fighting this way. Well, this family, since they're mostly left-handed, they built all their stairs the other way to give them the advantage to fight when people came and invaded their castle. So left-handed stairs, left-handed helices in here. Okay. If we look down on the helice, you actually see a little small area inside there. This is too small for anything to really fit into. So we typically see a lot of our hydrogens in here just kind of wrap around this. And you can see our residues kind of pointing out away from it. Space filling model looks like this. Here, if we do this kind of numerically, you can kind of watch these different protein amino acid side chains step down there. So I really do think of this as like a spiral staircase. Okay. So this is the pictorial stuff. A little more information about this. The top view, the inner diameter is about 4 to 5 angstroms. So that's really too small for anything to fit inside. When we get to our collagen triple helix, we'll see how that's formed. Because it's made of 3 of these helices, the inside is actually a little bit bigger. And hydrogens, turns out the hydrogen from glycine can fit inside. So every 3rd residue in a collagen is a glycine, so that it will fit on the inside. But our regular alpha helix is too small for anything to fit in there. Outer diameter tends to be about 10 to 12 angstroms. So cool thing about this, that's about the width of the major groove of double-stranded DNA. So a lot of our proteins, you can think of like our histones in particular, are going to have alpha helices that can just kind of sit in that major groove and interact with DNA to help facilitate winding or unwinding or making it accessible, all sorts of that good stuff in there. And of course, if we look at our amino acids, number 1 and number 8 kind of sit almost on top of each other. So if I go back up, you can kind of see this. Here's number 1, here's number 8, and there, if you're looking down on the helice, they almost line up. They're close to that. Really, I'd say it'd be like 1 in 36, but 37, but yeah, close enough. Okay, so good question to think about. What kind of sequence would give you an alpha helix with 1 hydrophobic face? We can actually have an amphipathic one where we put all the charged residues on one side of the staircase and all the nonpolar ones on the other side. So you have kind of an amphipathic helix. You have a charged face and an uncharged face, hydrophobic and hydrophilic. Okay. Not all polypeptide sequences will adopt an alpha helical structure. There are some residues, some amino acids that promote an alpha helix, and there are some that break it. Okay. So alanine and leucine are strong helix formers. If you have a protein sequence that has lots of alanines and leucines in it, probably a good chance it's going to be an alpha helix. Unless you see proline in here or glycine, because they will disrupt it. And they disrupt it in two different ways. Proline, remember, is a helix breaker because its R group goes back and attaches to the amino group. So it actually has that chain there. So it prevents it from adopting that alpha helical formula, because that psi angle, sorry, phi angle is made impossible, okay, due to that bend of the proline. On the other hand, so proline is highly constrained. It can't get in that position. Glycine, on the other hand, it's wildly unconstrained. It will not adopt the alpha helix because there's so many other interesting positions it could be in. I'm going to be over here, I'm going to be over here, I'm going to be over here. Because glycine has that small, a tiny R group, it can support a lot of other different conformations. So it will also break the alpha helix for a completely different reason. Okay. Depending on your side chains, if you have attractive or repulsive interactions, so you've got two positive charges that are pretty close to each other, that's going to push them apart and disrupt alpha helical formation. You've got a positive charge and a negative charge, they're going to stick together and that will favor it. So there's lots of factors that go into it. I like this chart just because it has the delta delta G naught. All right, so it's our change in the change in free energy that we're looking at when we put these different things in here. So we kind of arbitrarily assigned alanine a value of 0 because if you have a sequence composed entirely of alanines, it's going to make an alpha helix, no trouble. So everything else is a little bit different. So when we talk about was it leucine, 0.79, arginine, all these, anything that's below 0 here, very, very favorable for that. You see something like asparagine, that's less favorable. We see a bigger change in energy there. So asparagine seems like, we can fit an alpha helix, but a whole strand of asparagines probably wouldn't make one. You look at glycine here, 4.6, that's our huge change in energy. There's so much other stuff we could do to constrain it into an alpha helix would be energetically unfavorable. Same way if you look at proline, all I can see here is it says greater than 4 because you can't force it to adopt this alpha helical structure because it just can't bend that way. It'd be like those jujitsu stuff. Just snap the arm. All right. Okay, good times, right? Okay, so you don't need to know any of these values or anything like that. You just do need to know that alanine and leucine are going to promote alpha helix, and glycine and proline will break it. All right. It might be a good idea to know why. Okay. We can also talk about dipole interactions here. So remember, our peptide bond has a negative pole and a positive pole in here due to that stabilized resonance structure where we kind of stabilize the CO minus and either the N plus or C plus in there. So all the peptide bonds are going to have a similar orientation. So we're essentially lining up all of these tiny little dipole moments so we can make a large macroscopic dipole moment. So our alpha helical protein, we might have a negative pole and a positive pole in there. We kind of act like magnets or good stuff in there. Right? So our negatively charged residues will often occur at the positive end of the helix dipole. We put them down there because they're more stabilized. And maybe our negatively charged ones would be and our positively charged ones would be at our negative end. Okay? So if we look at this visually, we can say here's our dipole moment. Our carboxy terminus tends to be the negative side, and our amino terminus the positive side. So we'll stick aspartate and glutamate down here, asparagine and glutamine up here. Okay. And again, that will help stabilize the alpha helix. Okay. So that's a lot about the alpha helix, right? Comfortable, that's one, right, of our secondary structures. Okay. We also have beta sheets. Okay. This is our other organized, non-random structuring here. And our beta sheets allow this, are formed due to the planar nature of the peptide bond. You can end up making these kind of like pleated sheets that kind of run back and forth and can stack on top of each other. So what you can do is if you have this sheet like this, you can have another sheet underneath And hydrogen bonds can interact with each other to stabilize it and kind of hold them in this particular structure. So we tend to draw these as little arrows. And we draw this to show the actual angles in there, but a lot of times we'll just draw this kind of straight arrow or maybe a twisting arrow or something like that, depending on how accurate we're trying to be. At this level, a lot of times we're not drawing particular structures here. We just start drawing cartoons or ribbon structures. We'll say, here's a few arrows to indicate the beta sheet. Here's a few loops to indicate the alpha sheet. Here's a little string to indicate the random coil. And then maybe we'll draw the specific functional group that we're interested in. All right, so the key is our side chains are going to protrude from the sheets, kind of poking up, poking down here. Okay. All right. So there's two types of beta sheets. I guess you could say there's two types of alpha helices, left-handed and right-handed, but we have parallel beta sheets and anti-parallel beta sheets. Or if you're like the engineering professor from when I was undergrad, you could have anti-parallel beta sheets and anti-anti-parallel beta sheets. Or anti-anti-anti-parallel. Anyway, it's funny to me. Okay. So if we have multiple beta strand interactions, we get these lovely little sheets in here. And I suppose, I should be more accurate. A single line is a beta strand. When you have multiple of those lines acting together, that's when you get your sheet. It's kind of like, one thread is just a thread. You weave a bunch of those threads together, you can actually get a sheet. Okay. So the sheets are held together by the hydrogen bonding of the amide and carbonyl groups from the opposite strands. We've got two major orientations. Parallel sheets, the amino to carboxy are all running in the same direction. So maybe they're all going left to right, left to right, left to right. So how do you connect those? Usually there's a big loop. We'll talk about motifs, but there's usually a big loop here. So this runs across this way, and then we'll have this big loop that'll connect down to the middle one, and it'll run across this way, and we'll have this big loop that'll connect down to the bottom one. Okay? And you can see for our parallel beta sheets, we have these hydrogen bond interactions here. Note that our hydrogen bonds are at an angle. Okay? When we compare this to our anti-parallel, they're going to line up. So anti-parallel will be a bit stronger than our parallel. But our hydrogen bonds, again, are between, the key here is they're between the carboxylic acid and the amino groups. Okay. All right. So these are bent. So our pi bonding is not linear. It's not stacking as nicely as if we look at our anti-parallel sheet. Here we have this one go across this way, then connects down here and runs back this way, and connects down here, runs back that way. And you can see in our anti-parallel, our carboxylic acid and amino groups line up in a nice straight line. So we get a slightly stronger hydrogen bonding interaction in here. So anti-parallel is a little bit harder to tear apart. We good? Yeah, so just like taking it all in, like, all right. I'm really toning down I'm trying to cut down on my caffeine, because if I get going, I'll start talking like the Micro Machine guy, and I'm old enough now. You guys are like, who? So it used to be this commercial for like little tiny toy cars. Because they're so tiny, that the spokesman would talk real fast, and he just go on there super, super fast. And I'll get all hopped up on my legal speed here and talk really fast. So I'm trying to ease it back in. Again, we can see dimensions in here. I'm not going to ask a whole lot about dimensions here. I'm going to ask you more details about what's the difference between a parallel and an anti-parallel beta sheet. Talk about the hydrogen bonds in here. How are they formed? Okay. So with our anti-parallel, connecting the different strands can be a challenge, because you have to have this big loop that goes back. When we talk about motifs, sometimes you'll see an alpha helix loop that'll connect them, so you can have these barrels. Sometimes it'll just be some random coil. But for our anti-parallel, I can just turn these around here pretty quick. I'll put on my drifting tires and right around the corner there to flip around 180 degrees, or you can do a Rockford turn. Oh, what's terminology? I was a little kid. I got to stay home from school when I was sick, and I'd watch the Rockford Files with Jim Rockford, 70s something. Anyway, but there's actually a turn named around him, because so many times in the show, he'd flip his car in reverse, get going real fast to back away from people, and slam on the brakes while popping it out of the gear, twisting the wheel, turn around the other way, and then pop it back into gear, speed off. So the quick turn. So that's what we've got going on here. Quick turn, quick turn. All right, so our beta turns are going to occur frequently in our anti-parallel sheets. We don't have to, we could have a big long connection there and have this, but a lot of times for these anti-parallel beta sheets, we'll see this rapid turn, 180-degree turn. And it's going to occur over a 4 amino acid span. So these 4 amino acids are not part of the beta sheet, but they're part of the turn that connects two anti-parallel strands. Okay? There are two types of turns that are involved here. There's a type 1 turn and there's a type 2 turn. All right, and depending on which is which, you'll either have proline in position number 2 or glycine in position number 3. Like, aha, you know, proline and glycine break alpha helices, but they're awesome for beta turns because of that, the allowable angles of rotation. All right, so if we look at our type 1 beta turn, type 1 is our proline. So here we have, you know, one beta sheet. We have an amino acid here. Position 2 is a proline, and this constrained configuration in here. This is actually probably going to be a cis configuration as well too. Something like 99.96% of prolines are in the trans form. But when you get to your beta sheet turns in here, you suddenly see a huge increase, you know, like something like 5 or 6% end up in the cis form, so that we can help facilitate this turn. You can also see that the amino acids in 1 and 4 in type 1 form a hydrogen bond for an additional stabilization for that quick turn. Okay. Right. So if you're like me and have an all-wheel drive vehicle, you'll never go drifting because, well, yeah, unless you're on ice or something like that, because your wheels won't allow it. If you've got like a real-wheel drive vehicle, then you can, you know, drift around there and, you know, ball tires and stuff. Yes? So what we've got going on, let me go back up. Whoops. There we go, to right here. So what I want to do is I want to connect this strand to this one going in the opposite direction. So to do that, I'm making a turn. Okay. So what I've got, let's see, should probably flip these around. When I drop down to here, I'm actually taking that one strand, okay, and connecting around here to turn around. So this is my little turnout. Okay. Does that make sense? So here would be like beta strand 1, and then I'd have this turn and connect to beta strand 2, and then maybe turn back up beta strand 3, 4. Does that help? Okay. So type 1 utilizes proline. Proline in that cis configuration, tightly constrained, helps pull this around, and you have this stabilization here. Type 2 utilizes glycine in position 3. Okay? The difference here is, you know, glycine is kind of the opposite of proline because it's got this huge amount of freedom. Glycine is super flexible, so it just kind of rotates back and spins over here. Thinking about, what was it? You go back to The Matrix, I'm like, you know what, that movie's so old now. I know. And there was only ever one of them, but you got the part where, you know, Neo's dodging the bullets and leans back and balances up on one foot. Or if you've ever seen acrobats, you know, that can do these kind of seemingly impossible balancing things. That's essentially what our glycine is doing. It's bending in ways that the other 19 amino acids can't bend because of those phi and psi angles. And again, you know, that rotates back over on itself. We see an additional layer of stabilization here with another hydrogen bond that gets formed. And our type 2 is another way we can make that turn. Okay. So in both cases, we're just trying to connect these two opposite direction beta strands as tightly as possible. Okay. Okay. So let's talk about that proline just a little bit. Most peptide bonds that don't involve proline are in the trans configuration, but for peptide bonds that do involve proline, about 6% of them are in the cis configuration, and most of that is that beta turn. So we actually put them in this energetically unfavorable, you know, cis configuration to facilitate that turn. And then you can see all those hydrogen bonds between the strands there are going to pay the energy debt for putting proline in that cis configuration. Okay. So you can, you know, one of the ways you can think about this, which gets a little probably too crazy, is in terms of paying for the energy cost of getting rid of entropy. I think sometimes that can be helpful, but it kind of depends on the way your brain works. A lot of times I just completely ignore that and don't worry about it. Whatever strategy makes the most sense for you, that helps you make sense of it. Because we can come at biochemistry from like 4 or 5 different angles. All right. Pictorially, this would be our trans configuration. You can see, we have our alpha carbon over here, carbonyl group over here. So they're kind of across from each other from this. Our cis configuration, we kind of wrap these around so that this is kind of rotated around this way. Everything's kind of pointing together. So again, you can see this is a little more steric hindrance here. So we'd have more, energetically that'd be less favorable. But if we put that in our turn, you can also see, it's kind of like if you're running down the hallway and you grab a hold of the pole to help you spin around and make the turn. That's facilitating that turn. So, all right. I always hesitate when I talk about this stuff because, with biochemistry, every little section you can go deeper and deeper and deeper. And even though this isn't, so this is an upper level undergrad course, but it's almost like an introductory course to, the depth of biochem. So I always hope to get more like biochem majors so we can poke into this, but I try and cover a little bit of everything. Sometimes, I maybe get a little too deep in some areas, or hopefully more of that than not deep enough in other areas. All right. So how do we figure out if something is an alpha helix or a beta sheet or a random coil? All right. We can look at absorption, okay? Because if we start making these hydrogen bonds and we start having these things in particular configurations, that's going to affect how light gets absorbed. So we can look at spectral data and kind of assign values based on absorption at particular wavelengths. Okay? So circular dichroism is one method. I'm always amused when I talk about circular dichroism because that was the one part of my graduate work that did not go well. We didn't actually have a CD machine at our university. We had to go to Pittsburgh to borrow one. And so we'd spend all this time prepping, getting our samples ready, all that, drive up in my research assistant professor's minivan, which was an inexperience. And we get there and I get my data and my signal to noise ratio was 1. So basically I had all this noise, then I had my signal that was about the same as my noise. So I could never quite make that work. But circular dichroism ideally will look at the absorbance of light at particular, basically polarized light at particular wavelengths in here. And we can calculate the difference in this in the molar absorption to kind of give us an idea of, match that up with our alpha helices, beta sheets, and that stuff like that. So because of the chirality of these compounds, that's how we're gonna affect the absorption in there. So what we can basically see is our alpha helice, we'll see this lovely little peak somewhere around 190. Our beta conformation will be like 195 to 205. And our random coil, our peak would be about 215. All right. You can still see we tend to measure the beta conformation over here at about 205 because that gets rid of any residual absorbance from the alpha helix. Okay. So if we look at that absorption value, we can come up with a kind of map for different little regions of the protein. Okay. So for secondary structure, again, the big thing is alpha helix, beta sheets, and a random coil. Random coil is the other stuff. Our alpha helix, just be able to tell me about it, the relative sizes and how many residues per turn and all that good stuff in there. Our beta sheets, you've got parallel and anti-parallel. For anti-parallel, you've got 2 types of turn, type 1 and type 2, that involve proline and glycine respectively. Okay. For tertiary structure, we now starting to look at the big picture. We take one whole peptide strand and maybe one section of it is alpha helix and another section is beta sheets and another section is random coil. But we're going to see how it all fits together overall in a 3-dimensional structure. Okay. So again, our tertiary structure is going to be stabilized by just hundreds, if not thousands, of weak interactions, possibly even, well, or hundreds of thousands of weak interactions. Lots of these will be hydrophobic interactions. Lots of them will be polar interactions. We'll often see disulfide bonds that will kind of provide like an anchor to hold a couple different strands together. A lot of the amino acids that interact with each other might be really far away. We might have one at like position 10 and one at position 300, but it loops around and ends up close to that amino acid at position 10. And that's how it interacts in here. We can divide up our proteins into two major classes: fibrous proteins, so they're going to be like fibers, and globular proteins, which will be big blobs that tend to be water or lipid soluble. A lot of our fibrous proteins are insoluble or hydrophobic. So if we look at these visually, here we see a collagen, which has this triple helix. We basically have these three alpha helices here that then wrap around each other. And then here we have a this is looking edge on down into this. So this is our collagen fiber. One of the neat things about collagen is it's got a repeating protein sequence. So all these alpha helices will have GXY, that'll repeat, where G is glycine, and then X and Y are two other proteins in there. Probably shouldn't say Y, because that's what, tyrosine? No, but GX1 and X2. All right. Our fibrous proteins, we'll talk about a little bit, just a little bit in this chapter and a tiny bit in chapter 5 as well too. But typically they tend to be tough, insoluble, protective. If they're in the beta conformation, they might be soft and flexible, like spider silk. You have your collagen triple helix, which has a lot of tensile strength, but it doesn't stretch. So if you think of like your joints in your knees, your cartilage, and all that good stuff in there, that can provide flexible structural, strong but flexible structures in there. All right. So if I look here, we've got here a cross section of a hair. So we'd be looking at like keratin. Here are different cells. We have these intermediate filaments, which are composed of protofibrils, which are composed of protofilaments, which are composed of two coiled alpha helices. So you basically take one alpha helix, you wind that around another alpha helix to get your protofilament. So your coiled coil, you wrap a few of those around each other to get your protofilament. You wrap 2 or 3 of those around each other to get your protofibril, and so on. And you just basically kind of build it up. You guys ever like have a burst tire, like you see like the wires that are inside your tire to give it strength? You'll have, you look at it, you have like one little wire there, but you might be wrapped around another wire, and you take a couple of those and wrap around each other, and you put those in some plastic and then wrap those around each other, and then you weave those together to give the strength to your tire. Essentially the same thing is happening here with our fibers. If we talk about keratin in hair, we look at alpha keratins. Here again, we have our two coiled coil chain here, alpha helix wrapped around an alpha helix. We take a bunch of these, so you have the end regions here. Our end regions can interact with each other, but they're offset, so we can wrap a couple of these coiled coils around each other to make our protofilaments. We can take a bunch of protofilaments and wrap those around each other to make our protofibrils, and we end up with a hair fiber. Okay? So we take these microscopic structures and just kind of like, I always think of like Rumpelstiltskin. Weave me some hair. Okay. So speaking of hair, always a fun topic here. If you ever get your hair permed or relaxed, you're essentially breaking these disulfide bonds. So everybody's hair has a particular genetic structure in here. The genetic imprints say, hey, make these disulfide bonds. So in this case, maybe we've got somebody who's got straight hair. They don't want to have straight hair, they want to have curly hair. So you apply a reducing agent that breaks these disulfide bonds. Okay, so you have these here, you wrap it into the shape that you want. So if you're straightening it, whatever that, if you're curling it, you wrap it around the rods or whatever there. So you get it in the right shape, and then you add an oxidizing agent to make these new disulfide bonds, and the hair is now permanently straight or curled, depending on which way you're going. So you can change the shape of it that way. And it'll stay that way because you've actually done some chemistry here and changed the connecting the structure based on the connections of these disulfide bonds. As new hair grows, it'll go back to the genetic pattern. So, but this will be changed, whatever. Yeah. So reduce.
I have a question.
Put in as you think. Yes.
So what if we're doing reverse, like curly to straight? And what happens when we actually get water on it and it goes back to curly? Can you explain that? Just curiosity.
Ooh, that's a good question. I would suspect that's probably more of a water being absorbed, but I'll have to look into that. I don't think the water is actually going to be reducing those new bonds that you've made to take it back to that way. But I would suspect, because you'll see people who, like, in high humidity as well too, when their hair kind of frizzes out. I think it's more the ability that hair can have to kind of absorb that water, and that absorption kind of forces it into a curly nature.
Okay.
Ideally, if you've straightened your hair and you get it wet and it kind of curls up, when it dries, it should mostly go back. But then again, you might not be able to completely dry the hair fibers. But I'll look into that and come up with a more detailed response for.
Alrighty, thank you.
You're welcome. Yes.
Okay, so how does it work? Like, because people who tend to have straighter hair when it's curly, so that like the boss will be breaking thread.
Yes. Yeah.
But for people who have curly hair when they perm it, it's straightened.
Yes.
The boss still breaking thread?
Yes. Yep. So you're basically just kind of starting over here. So you have this curly hair that's held in this more angled position. Okay. So again, you reduce this, break those bonds, and kind of stretch it out so that you're keeping it straight. I guess, is that where they apply like the foil? We're getting out of my area here. I don't spend a lot of time at the salon for dyeing hair. Okay, for the foil. Yeah. I know there's something you do to like straighten it out, but when you do it that way, you hold it in the straight position, and then you apply the oxidizing agent to make those new bonds and keep the hair straight. And the key is just kind of have that hair stretched out and straight in the position that you want. Whew. Yeah, I need to spend more time at the salon. All right, yeah. But again, the chemistry of hair, it's funny because there's a friend of mine in New York City who, I think she modeled when she was younger, and she's big into like the beauty products and stuff like that. So I guess one of the anti-aging thing that's come out is keratin. So applying keratin particularly as a kind of a filler for wrinkles under the eyes. So I remember one time on Facebook, she's like, I love keratin. Well, my actual area of research is using human hair keratin to, try and get peripheral nerve cells to regrow. So I'm like, I love keratin too, probably for different reasons, but so multi functions here. All right, there we go. I need to expand that slide apparently. All right. So I think I've got maybe one more fibrous protein to talk about, and that's collagen. And it's funny because, I like talking about keratin more now because, that's what I research on. But collagen was the thing that kind of got me interested in fibrous proteins because it's so kind of neat. It's a big constituent of our connective tissue, tendons, cartilage, bones, even the cornea of the eye. So we have this long glycine proline-rich left-handed helix. So there's lots of prolines in here, even though we got alpha helices in here. So we take these three collagen chains and intertwine them into a right-handed superhelical triple helix. All right. It's not just a helix, it's a triple helix. It's got a really high tensile strength, so it's stronger than steel, so you can go out and like punch buildings and knock them down. No, okay. But if you get a bunch of these triple helices, they will assemble into a fiber, collagen fiber. And again, we're kind of looking at this, looking down on here, we'll see all of our glycines are pointed to the middle because our left-handed triple helix has enough space in there for those glycine residues. So the glycine residues all point to the middle. Okay. All right. We do see one of our modified amino acids show up a lot in collagen, this 4-hydroxyproline. So basically, by adding this hydroxyl group to proline, that allows it to adopt a more favorable pucker conformation, which will allow these collagen helices to form more easily. There is some post-translational processing, something we just kind of touch on this semester, but we'll talk a bit in more detail on in second semester biochem. But our post-translational processing, we have this prolyl hydroxylase enzyme. We're also going to see a cofactor of alpha-ketoglutarate. We're going to require oxygen and vitamin C. All right. So when we do have all those things, our prolyl hydroxylase will hydroxylate our proline here. It's in the endo form normally, but when we put the hydroxyl group on there, if it's in the endo form, that hydroxyl group could interfere with this oxygen sterically. So we put it in the exo form, so it's more favorable. Okay. So collagen, important for skin structure. Yeah. One of the things they look at for like older people, you know, you go to the doctor and kind of like push on the skin and see how quickly it bounces back, right? What happens if you don't get enough vitamin C? Any diseases you might think of? Scurvy, exactly right. Why British sailors are called limeys, because you know they used to put citrus limes so they would get vitamin C. Because if you don't have enough vitamin C, then your prolyl hydroxylase ends up being less active, downregulated. You don't have that cofactor in there, so you can't make this collagen. So people would get scurvy, and like their teeth would fall out. You know, it's like a big book of British smiles or whatever would stem from, you know, scurvy. So wait till we talk about rickets. All right. So why do we need this vitamin C? It's going to actually restore our iron 2 state. Okay. So we got to have our iron 2 in here. So our alpha ketoglutarate ascorbate to succinate and dehydroascorbate, allowing us to do our oxygen chemistry in there. All right, not that I'm going into any great detail about that, so I'll gloss over that and move forward here. Our collagen fibrils make these lovely little structures in here. You can see these kind of cross striations in here. These are various little head groups that will interact with each other. So as they wrap around and form each other, you get these nice little cross-linking between them. So our disulfide bonds are cross-linking our various fibers together. Covalent bonds between lysines and hydroxylysine and stuff like that. Not disulfide bonds, but the cross-linking that way that helps stabilize them so we can have our strength. The last fibrous one is silk fibrin. I always forget about that. Want to collect a bunch of the spiders that live outside my house and make a suit of silk out of them, right? Probably the wrong type of spiders there. But fibrin is the main silk protein, as opposed to alpha helices. The reason we talk about this one is because it's an anti-parallel beta sheet structure. Okay? So the small side chains, alanine and glycine, allow these to be packed very closely together. So you get a lot of this close quarters hydrogen bonding. And you also have these London dispersion forces, because they're so close to each other, that allows a very stable and strong interaction between the two of them. So that's why spider silk is so strong. And if you think about it, if you're walking to a spider web and it's like, hey, you can't tear it to get it off your face. Ah. I'm sure that's going to look great on Zoom later on. Play that over and over again. I'm going to be a meme. Only way I'll be famous on the internet. All right. So we see this very close packing of this anti-parallel sheets in here. You get multiple sheets that interact with each other, and you end up with this very strong material. On an atomic scale, silk fibers are stronger than steel. They're also very flexible. They can stretch a lot before breaking. So again, your spider web, you walk into it, you got a lot of flex in there. It's very composite. You have these crystalline parts, which are fibrin rich, and you have some rubber-like stretchy parts as well too. Stretchy, hang on the body. Oh, yeah, random things from my past. All right. When we next pick up, we'll start talking about water-soluble globular proteins, but I think this is a good stopping point. I doubt I'll see too many questions about this chapter yet, because we got chapters 1 through 3. My plan for Tuesday is to do a kind of review session, and hopefully by Tuesday I will actually have your exam made up. I have some exams. I'm like, all right, how am I going to do this? Yeah, what am I going to do? What am I going to do? As far as, oops, get rid of this. As far as submitting stuff, like say for lab, all right. Let's get rid of this. All right, and pull up this. Yes. All right, let me share the screen again. Okay, go into Biochem 1 lab. All right, maybe. Take a look at this. Go into, I hope this will work this way. If I go to modules, okay, scroll down here to, you might have to go into assignments. It's a test. Yes, if you scroll down to assignments under lab and click on the lab, it will allow a file upload. So if I show you guys here, right, there should be, yeah, a way you can upload the file. Yes. Uh-huh.
Write out the map?
Yes, yeah. If you want to write it out and then scan it and send it back in, yeah, that'll be fine. Yeah. Yes.
Did you give us numbers? Or
Yes, yeah. Did you got them? Okay, good, yeah. I think I got a picture somewhere.
Do we need to submit the pre-lab questions on the first part of the lab?
No need to submit the pre-lab. What was your question?
What file was to submit?
So generally, PDF, Word, anything is good there too. I'm not restricting it, but
And where do we find them in?
so if you go under assignments, and then scroll down, it will say labs. Okay, and then you'll be able to click on that lab. Okay, and then the other important thing I should let everybody know is, because I know the first time I always forget to do this. Well, depending on how lab reports go, I've been grading them out at 25 points because I had this rubric for written lab reports. Still waiting to hear more from Labster, but everything's out of like 25. So if you get like a 23, it's like, oh my God, how did I fail this lab? No, it's everything will be out of 25 points.
We only have to submit this sheet, not
Yes.
the lab report.
Right, correct, right. No big lab report for this one. So this is just kind of the worksheet type deal going on here.
And you said for our numbers, that's like the error.
So you can just leave it in milligrams and you should be okay. Yeah. Okay. So I think that's everything. All right. Any